Hello, friends. Good morning to all of you. And again, I am glad to share some of the thoughts and the understanding in cancer biology. In fact, this particular topic, which is highly understood and as well as debatable, what is the role of lactate in cancer growth and proliferation? If you look at my dear friend, this lactate is a well-known metabolic product within the cancer cell as well as other healthy cells like muscle cells, and that is known, and this lactate production in the context of cancer cells has gathered attention in the recent as well as several decades. What if the lactate is produced inside the cancer cell? What is the destination of this lactate metabolic product? How it participates in various cellular regulations, including metabolic regulations or epigenetic regulations, and many more. So I thought I should discuss one of the aspects of lactate in the context of tumor microenvironment, if you look at here. So obviously, what I'm going to explain here, lactate, it is considered very long period of time as a metabolic waste in cancer cells. But if you look at why the cancer cells, they will produce large amount of lactate within the cell. Definitely one question is always there, okay, during the production of lactate from the pyruvate through the glycolysis pathway, the cancer cells may utilize more amount of glucose and at the same time rapid ATP production is achieved. But the story is beyond that, the requirement of the cancer cells for the ATP, but maybe cancer cells are having strategies beyond just ATP requirement. So what I propose here, lactate definitely maybe as a metabolic waste, but interestingly, it turns out to be as a metabolic weld within the tumor microenvironment. Whenever I say here metabolic weld, it does not mean only for the cancer cells within the tumor microenvironment. Instead of, these lactate may serve as a metabolic messenger or the weld for non-cancer cells within the tumor microenvironment. And these non-cancer cells may be referred here pro-tumor like immune cells, stromal cells that support cancer cells to proliferate, to grow, and to go for various other tumor hallmarks. And most important, the component of tumor microenvironment that can use lactate as a metabolic well, that is microbiota. So microbiota, the presence within the tumor microenvironment, they may also utilize lactate produced by the cancer cells to generate certain byproducts or the products, and in turn these products may help cancer cells finally for their growth and proliferation. That is the whole story. Okay, so in that direction, I would like to share the proposed model here. So what is the proposed model here? If you look at in the tumor microenvironment, there are cancer cells, obviously. So like this is our cancer cells. In the tumor microenvironment, if you look at the microbiotas, that is one of the component of tumor microenvironment. And then if you consider the pro-tumor non-cancer cells, that can be in the form of macrophages, that is the immune cells. And I will explain how the lactate as a metabolic waste is going to be used as a or proposed as a metabolic wealth for not only cancer cell but also non-cancer cells within the tumor microenvironment. But these non-cancer cells, including immune cells or microbiotas, they are friends of cancer cells. They are pro-tumor. They support the cancer cells to achieve their malignancy, to achieve their tumor hallmarks, etc. Okay, so we'll start from here. If you look at, my dear friend, this is the cancer cells, and as I said, through the glycolysis, pyruvate is going to be generated from the glucose through the glycolysis pathway. That's what we know. And once the pyruvate is generated, most of the time, pyruvate is going to be taken up inside the mitochondria for the routine oxidative phosphorylation, or you can say the tricarboxylic acid cycle pathway for the ATP production. That's what we know. But what is happening here, the alternate pathway or the existence within the cancer cell as it is proposed, that pyruvate is also converted or into two metabolic substrate. One is the acetate. So the pyruvate can be directly converted also into the acetate, and pyruvate is also going to be converted into the lactate by one of the very famous enzyme, that is lactate dehydrogenase. This one. Once the lactate is generated, that is the whole question mark. What is the destination of this lactate? How it is going to impact the cancer cell landscape, and also how it is going to impact non-cancer cell landscape, including immune cell or microbiotas. So look at here. So what we are, there are certain evidences that, okay, lactate is going to contribute for one of the recent discovery, that is lactylation process here within the nucleus, and that impact the epigenetic or chromatin state of the cell, and ultimately that is going to be you can say playing a role for cancer cell as well as also the non-cancer cell like immune cells. But the whole problem in the field is lactate, once it is generated, how the lactate is going to be converted into the lactyl CoA, this one is a debatable question. There is no any evidence on which enzyme is going to contribute for the conversion of lactate to the lactyl CoA. Still, there is also doubt and debate whether the enzyme that will convert lactate to the lactyl CoA is expressed within the cancer cell. That is also a debatable question. Another very important point is, for the lactate conversion to the lactyl CoA, the required coenzyme A, that is very, very famous cofactor for various metabolic enzyme, epigenetic enzyme, like epigenetic acetylation process. This CoA is also going to be utilized by the lactate for the conversion to the lactyl CoA. So that means CoA pool inside the cytoplasm, and that CoA as a cofactor is very, very important for normal cellular functioning, is also going to be channelized from this direction to that direction. That means the possibilities are there. Cancer cell is going to divert the pool of CoA from acetate to the lactate, and then lactyl CoA is going to be generated. Great. And this lactyl CoA potentially may impact some of the epigenetic enzyme for lactylation for epigenetic modulation. Look at here, this way. But the question is there, where is the enzyme? This one. There is no any evidences. There are certain enzymes that convert acetate to acetyl CoA, that is acetyl CoA synthetase. So that is reported. So from acetate to acetyl CoA, the enzymes are reported, but for lactate to lactyl CoA, enzymes are not reported. That is the gap in the field. Fine. Now, if we have the evidence of lactyl CoA is getting inside the nucleus and then they perform lactylation, but another gap in the field is the epigenetic enzyme that is going to perform lactylation by using lactyl CoA still is not confirmed, and there is some debatable questions. What is the exact enzyme that perform this epigenetic process, that is lactylation? But if you look at whether the enzyme is not reported or discovered, irrespective of that, if you look at this whole process, the conversion of pyruvate to the lactate, then lactate to the lactyl CoA, and then lactyl CoA is going to impact the epigenetic landscape. Whole pathway is pro-tumor. It is helping the tumors or cancer cells to achieve the hypoacetylation, or you can say it works in favor of cancer cell for their growth and proliferation. So irrespective of the lack of evidences on the participating enzyme, but the possibility of this metabolic link between lactate production to the epigenetic modification is also convincing. But in future, these enzymes has to be discovered. Fine. Another thought process is there, whether cancer cell, because the production of pyruvate is more, and pyruvate is going to be converted into the lactate. So once the lactate is generated in excess amount within the cancer cell, for the cancer cell it becomes, or it may become, as a metabolic waste. Some fraction of the lactate can be utilized by the cancer cell for lactyl CoA, this pathway, and then epigenetic modification. But majority of the lactate, their destination, what is the role? That may be a debatable question. So what is the possibility here? If you look at this lactate may come out of cancer cells in the tumor microenvironment. This lactate can be accepted by pro-tumor microbiotas because lactate is more. Cancer cell is not interested to utilize all the lactate produced within the cell. So the lactate is going to be sent outside of the cancer cell. And there is also very well clear evidences. There are transporters within the cancer cells on the membrane of cancer cell that can, you can say, sent outside these overabundant lactate to the tumor microenvironment. That is the situation. Look at it. Once they are sent outside lactate, the possibilities are there. Some of the pro-tumor microbiotas, they have the mechanism to convert lactate to the lactyl CoA. So the possibilities of contribution by the microbiotas within the tumor microenvironment, possibilities are there, but still it is not well supported by the evidences. Fine. So whatever way, whether lactyl CoA that is generated within the cancer cell, they can make exit from the cancer cell to the outside, or lactate is going to come outside of the cancer cell, converted into the lactyl CoA. This pool of lactyl CoA is going to be present within the tumor microenvironment. So here is the two options. One is the lactyl CoA inside the cancer cell. They are contributing for or helping the cancer cell to achieve certain epigenetic state in favor of cancer cell. Other possibilities are there. Lactyl CoA within the tumor microenvironment outside of the cell, once the level reaches to an extent, this lactyl CoA may have some pro-tumor activities. And what is the pro-tumor activities is suggested in this way. This lactile or pool of lactile CoA may be received by anti-tumor, or that is M1 macrophages. Look at here. And these anti-tumor M1 macrophages, once they receive the metabolic messenger in the form of lactile CoA, these M1 macrophages are activated. They turns into in favor of cancer. Look at here. So this M1 macrophages is going to go for polarization, and they are going to turn into M2 macrophages. So what is, see the link here? Lactate produced by the cancer cell here in high amounts, lactate and their product lactile CoA, they are going to be channelized outside of the cancer cell to be given as a metabolic messenger to the non-cancer cells, either microbiotas here or anti-tumor M1 macrophages, so that the anti-tumor macrophages is going to be turned into the pro-tumor. That is one of the beautiful example of how the cancer cells are going to convince or manipulate non-cancer cells that should be able to stop cancer cells or kill cancer cell, but ultimately they turns into pro-tumor. Look at here. So it looks very wonderful strategy by the cancer cells utilizing the, it appears like a metabolic waste that is in the form of lactate here. This one, if you look at, yeah. So this lactate appears a metabolic waste, but how cancer cells are going to utilize these metabolic waste as a metabolic wealth or the messenger to convince non-cancer cells, including microbiota or macrophages, to turn in their favor. Outstanding. And that is why, in a very simple way, we can conclude, yes, tumor microenvironment is so heterogeneous and complex. There is not a one type of only cancer cells. There are several heterogeneities are there. Several type of non-cancer cells are there, and all together they make a team of pro-tumor, pro-cancer microenvironment. So what we learn from these studies, if you look at here, and all these explanations, it leaves certain biological questions. And what is the question here? If you look at the burning question that can be raised, whether metabolic waste, that is the lactate, produced within the cancer cell is going to be shared or transferred to the pro-tumor microbiota within the tumor microenvironment. Then the whole question is, why on the earth cancer cell will transfer this lactate or metabolic waste to the microbiota for a return gift? Look at here. For a return gift, because the cancer cell knows that whatever cells are in the surrounding, ultimately they will work in favor of cancer cells. They will support cancer cell one way or either way. So that is why the cancer cell is very smart enough to share these the metabolic waste, it appears lactate, to the microbiota, so that microbiotas will generate lactile CoA, and these lactile CoA can be used by cancer cells or non-cancer cells for the desired epigenetic modification, and that is as lactylation. Great. Another question is, isn't it this, the sharing of lactate to the microbiota, then lactile CoA between macrophages to the cancer cells, etc., is an example of give and take. So the cancer cells are generating a lot of lactate as a waste, but ultimately this lactate is going to be used as a process of give and take. Cancer cell will give lactate, microbiotas and macrophages, they will turn into lactile CoA, Then ultimately, finally, cancer cells will get, return gift in the form of various chemical signals from the pro-tumor macrophages for growth and proliferation. And all these questions, still many of the experimental evidences has to be resolved. And in my lab, in our lab, we are also addressing some of the question: what is the potential enzymes for epigenetic modification? What is the potential enzyme that can convert lactate to the lactyl CoA, etc.? And we hope that some of the interesting facts will come out. Thank you very much for listening, and I hope that this will help you to understand one of the aspects of cancer biology. Thank you very much. Keep using mask and be safe at your home. Thank you very much.